Okay, so this is a continuation of our discussion on digestion and absorption. We talked about the mouth and, of course, the fact that you start masticating food there. And that's where salivary glands come in and produce digestive enzymes such as amylases and lipases that begin to digest fat, at least minimally, and there's no absorption that takes place there, unless it's something like alcohol, perhaps. From there, you swallow it, and it goes down your esophagus, and I'll show some animations of this a little bit later, but the peristaltic movement is what causes the food to travel down the esophagus. We talked about the different layers, and again, I'm going to try to emphasize some of this by some videos that I've found. That food will then ultimately enter through the lower esophageal sphincter into the stomach, where you have your acids building up from parietal cells. This included things like the hydrochloric acid, as well as pepsinogen, that then gets converted to pepsin. And so your stomach will continue churning, kind of like a mixer, breaking up the particles smaller and smaller until they get quite small, so that it can actually pass through the pyloric valve and pyloric sphincter. There it'll enter into the small intestine, and that's kind of where we're getting at. But for the most part, there's some lipid-soluble substances like alcohol and aspirin being absorbed. But for the most part, it's just some more digestion, particularly of proteins and fats, and denaturing proteins, which means breaking down hydrogen bonds and making it a more linear structure. The small intestine is where we're going to continue on this discussion. I may briefly show you some animation of what's happening in the mouth and the esophagus and stomach, and then we'll move into the small intestine. Again, I'll try to show a little bit of animation as we go through this. But in the small intestine, this is where the acids are from the stomach, which would be called chyme at this point. The acids in the broken down food is called chyme, and it gets squirted into the small intestine. It will be neutralized thanks to the pancreas as well as the small intestine providing providing one second. Providing biocarbonate. I was having a little brain freeze for a second, which is HCO3. Remember, your body can make this biocarbonate through breathing in oxygen, and then you start breaking down sugars in your mitochondria and so forth. You'll get carbon dioxide buildup in your blood, which gets converted to bicarbonate. So this is bicarbonate is readily available throughout your body through those carbonic anhydrase, the enzyme that does that. And so, again, your body's really good at making this bicarbonate that's floating in your blood and so forth. So the pancreas will secrete this along with the cells of your small intestine. There's bile also coming in from the liver. Bile, remember, is going to be the breakdown of blood through when your blood cells and the hemoglobin starts to live out its lifespan. Some of that hemoglobin can be broken down into bile salts. So bilirubin is part of that. And that's going to be really important for fat absorption, because what happens is these bile salts will surround a droplet of fat and making it smaller droplets of fat. You'll see that as we continue in this lecture. So that's what's happening in the small intestine. That's the motility of it. Goblet cells are releasing mucus that again will provide protection to the small intestines, as long with hormones and things like that influence the rate in which these nutrients are absorbed and moved through your small intestine. So a diet heavy in proteins and fats will actually move slower through the digestive system than something like simple carbohydrates. So there's all that's kind of being regulated again by the hormones as well as the brain of the stomach, so to speak, that I talked about in the previous lecture. Peptides will be absorbed by active transport. That means it's going to use ATP along with amino acids. Glucose and fructose by secondary active transport, which include things like endocytosis. Water will be absorbed by osmosis. Fats can dissolve right through the cell membrane, so that'll be simple diffusion. Ions and vitamins can be transported in by active transport again, requiring ATP. So the small intestine is really probably the most important portion of your digestive system. Not to say that the whole thing isn't important, but this is where the absorption really takes place for water along with all the nutrients. Now, the large intestine will also absorb water as well, and this is where ultimately what wasn't absorbed in the small intestine gets made into food. There'll be segmental mixing going on throughout this whole process. We're kind of back and forth, whether we're talking about the small intestine or the large intestine or the stomach. There'll be kind of this churning action that is slowly propelling the food down the intestine. There's going to be goblet cells that are going to help again protect. A mucus layer will help protect the tissues with helping to form the poop. And there's not really any digestion taking place except for what's happening from the bacteria, the symbiotic bacteria, that obviously causes gases and smells and all sorts of stuff from the intestine. You will absorb water and minerals and vitamins, and most vitamins are also produced by bacteria, like vitamin K. So you can see how in the esophagus, the food is traveling down by peristaltic contractions that ultimately just push the food quickly down the esophagus from the mouth to the stomach. But when we're talking about intestines, you see this kind of back and forth churning. Where you can see if you look at section B, where it says segmental contractions are responsible for mixing, you can see it's like 2 steps forward, 1 step back when it comes to the mixing of the food. So eventually, the movement is very slow moving forward, but there's a lot of going back and forth. Now, the small intestine is actually quite a bit different in structure, even though the basic structures throughout the entire digestive system are there, like the mucosal layer and the submucosal layer. The difference, though, is in the small intestine, like in the stomach, you saw kind of ridges and grooves. Well, in the small intestine, you have finger-like projections. And these finger-like structures are called villi. And on these villi are what we call brush border cells or absorptive cells. That's what is listed here in this picture. These are small cells with microvilli on it. So you have villi with finger-like projections, and then you have cells lining it with smaller finger-like projections where the nutrients are actually absorbed. Some of those nutrients will begin to be absorbed in the manners I've described through active transport or endocytosis or so forth. And then those nutrients will often be passed on into the capillaries that are inside the villi, or it also include lacteal ducts that are part of the lymphatic system. So they're part of the lymphatic capillary. So different things will be absorbed in these different vessels sometimes. And we'll get into all that as we continue in the lecture. So here's another picture of the small intestines. You can see the villi are sticking up, like finger-like projections again. In addition, there's also little pits called Cryptic Lumen, and these vary in size as well. If you're a baby, these cryptic lumen are actually more important for digestion. And I think that may be part of the reason why some food allergies are formed when you're younger because of the size of this cryptic lumen. If you want to know more, that'd be an easy thing to look up. But my understanding is the cryptic lumen is smaller in babies, and there's more absorption of larger macromolecules, like peanut proteins or something, and that could trigger. And one of the reasons why they recommend not feeding certain things to a baby until they're older because they're more likely to get a food allergy. But again, I'm not that might be something that'd be good for you to look up a little bit more. I haven't really studied it in detail, but that's my understanding off the top of my head. You can see along this villi, there's those absorptive cells. Again, we call those brush border cells. And then you can see there's also goblet cells that are important for making the mucus. In addition, you'll see there's a Prior's patch. This is loaded with immune cells that help protect your body from foreign invaders, which makes sense because remember, the inside of the lumen of your small intestine is the outside of your body. So it's kind of the outside, and then whatever gets absorbed is actually technically inside your body. You can see the smooth muscles are, again, going in opposite directions. You have circular muscles going in one direction and longitudinal muscles going in the opposite direction. Again, this maximizes churning of the food and making a more complete contraction. And again, it's this mixing is kind of a two-step forward, one step back kind of approach to slowly moving food through the small intestine. You can see the submucosal layers where the capillaries are located also. But here's some more pictures of that. So you can see there's a pretty big difference between like the lining of your stomach, which were bigger grooves, and we had our gastric glands and our lymph nodes and vessels are all kind of deeper below it. But the basic layers are still there. The rugula is in the stomach. But then you can see it's finger-like projections in the small intestine. So again, the chyme is what's going to be in the stomach, and that's going to include basically the food that you've eaten in water, along with the acids that are present in your stomach. So that chyme will be squirted into the small intestine through the duodenum. If you look at this picture here, this is the duodenum is. And from there, digestive enzymes are going to take place. They're going to come from the brush border cells, which are the intestinal cells, along with the pancreas right here. And then also the gallbladder, which is located under the liver, which is going to secrete the bile salts into the same region of the small intestine. So there's going to be little tubes coming from the gallbladder into the duodenum, along with the pancreas. And so this is showing you how bicarbonate is used to raise the pH of the chyme. Remember, the chyme is very acidic and can harm the cells underneath it. And so what happens is the bicarbonate is secreted into the lumen of the small intestine. That's again the hole that's going through. And the pancreas is going to do this along with the brush border cells. And as I mentioned before, when you break down nutrients like sugars from the mitochondria, carbon dioxide is built up. Some of that carbon dioxide you obviously breathe out. Some of it, you can see how it's closely associated with the capillaries, can enter into the pancreatic cell and be converted from carbon dioxide and water into bicarbonate using the enzyme carbonic anhydrase. That's what CA stands for. Carbonic anhydrase. So carbonic anhydrase makes bicarbonate. That bicarbonate is then transported out into the lumen, ultimately making a very acidic chyme into a more basic, ultimately neutral solution, 7.3-ish or so. That's important for a lot of the enzymes to work. Because as you know, if you had a low pH your own enzymes, your own digestive enzymes wouldn't work, which are made of proteins, because that low pH breaks hydrogen bonds and so forth. You can see that a chloride ion is actually being going to do this co-transport, or anti-transport, as it's called. The chloride will come into the cell, and then ultimately it will be secreted back out through the CFTR channel, which is cystic fibrosis something channel. The main thing about that is to realize that that Cl helps to pull sodium and water out into this lumen as well. This is important for making the mucus more watery. And so people with cystic fibrosis, unfortunately, this channel doesn't work properly. And when this channel doesn't work properly, again, just due to some mutations that cause the protein not to have the proper structure, that's what causes cystic fibrosis. The mucus doesn't loosen up and become more watery, causing all sorts of havoc in their body. It's not just in the intestine. This CFTR channel be found in the lungs and other places, making that mucus more watery. When the mucus doesn't get watery, it is more likely to have a bacterial infection, particularly if it's in the lungs. So that's what this chloride's doing. It's helping pull sodium and water across the barrier. Here you can see the carbon dioxide, and just to kind of look at the chemical reaction of carbon dioxide and water being joined together by carbonic acid into biocarbonate, which is shuttled out. And then notice that the hydrogen, which will make the acid, is anti-transported out into the interstitial fluid while sodium comes in. And sodium gets kicked back out, potassium comes back in. So this is a sodium-potassium complex. And then the sodium and chloride and potassium ultimately come in together, and then potassium leaks out. It's just interesting to see these transports going on, because you realize you've seen something like this again in nerves and muscles and all sorts of other cells as well. So you can see how the movement of these salts and so forth are important for physiology, whether we're talking about nerve impulses or whatnot. It also explains why salt mixture is so important. A lot of this salt movement, and sodium chlorides and stuff like that, also provides evidence for evolution, because remember, cells evolved in oceans. And so they needed to regulate salt quite a bit in those cells, right? So here we have something like where we have salt being regulated, and then through evolution and modifications of these physiology becomes nerve impulses, or it becomes something else. But here you can see how salts are involved. So anyway, this is what neutralizes the chyme that comes into the small intestine. The biocarbonate is released from the pancreatic cells and the intestinal cells. And so this is what this picture is showing you here. This is the tube coming off of the pancreas. It's going to secrete proenzymes, or zymogens is another term for that. Remember, ZYOMGEN. Well, these zymogens or proenzymes are going to be secreted along with the biocarbonate into the small intestine. Now, this is also true that the brush border cells will also be releasing some proenzymes as well, but they also have enzymes on their surface called enteropeptidases that can activate this proenzyme trypsinogen. Trypsinogen is the precursor to trypsin. Trypsin is a type of protease. Now remember, proteases are enzymes that digest proteins. So trypsinogen is a precursor to trypsin, and the enteropeptidase activates trypsin. That trypsin then can go on to activate other proenzymes. Some of them are proteases also, like chymotrypsinogen. Chymotrypsinogen becomes activated thanks to trypsin into chymotrypsin because it'll cleave a part of that proenzyme off, making the enzyme active. So you can almost think of enzymes again, like proteases, as a Pac-Man digesting proteins, just to keep it simple. So trypsin will activate chymotrypsinogen into chymotrypsin. That's a protease. That will also help to digest proteins. And then we have procarboxypeptidase, which is an enzyme, carboxypeptidase, which is another protease. So again, remember, proteases are enzymes that break down proteins. We also have a procolipase. That colipase will be activated by trypsin again to become an active lipase. Lipases are enzymes that break down lipids. So that's what that enzyme will do. The colipase will break down the lipids that you have taken in and eaten. And then we also have prophospholipase, which will be broken down or be converted by trypsin into phospholipase. Now, phospholipase will break down phospholipids. Remember, phospholipids are the major component of cell membranes, phospholipids. So all these enzymes are critical for breaking down whatever you've eaten or some other animals eaten into the basic components that will then be ultimately absorbed and turned into human protein or used for human cells like the phospholipids. So here's how the proteases work. You can see the chemistry of digestion proteins. And so basically proteins are enzymes. So all proteins can be, proteins in your body are either structural or enzymes and so forth. So proteases are going to break down long chains of proteins. Remember, proteins are made up of amino acids and they have peptide bonds between them. Hopefully you remember what an amino acid looks like. It has an amino group and a carboxyl group. And then they'll usually have a side chain that makes it whatever type of amino acid it is. Well, these amino acids, again, are joined by peptide bonds that are going to be, a peptide bond is an amine group and a carboxyl group that's joined together. Hopefully, if you don't remember that, take a quick look in a online or biochemistry book or whatever, and you'll find how that works. So endopeptidases will break down these long strands of polypeptides, they call it a protein. They'll be broken down using water. And so the water will be used to break down a longer chain amino acid or peptide into smaller polypeptides. And then ectopeptidases will work on the external end. And so they will take an aminopeptidase and break it from the outer end. So that's what aminopeptidase does. And the carboxylpeptidase will break it from the other end, from the carboxyl side. So the amino side is the NH2, so the amino acid, or excuse me, the aminopeptidase will break it down again with water. That's why you got to drink so much when you eat. And that will break it down into their smaller and smaller amino acids. So that's what endopeptidases do and exopeptidases do. From there, these proteins can be absorbed. If they're the smaller peptides, thanks to the breakdown of these trypsins and chymotrypsins and the exopeptidases and endopeptidases, they'll be broken into these smaller and smaller amino acids. So di and tripeptides, which mean 2 amino acids or 3 amino acids, ultimately, can be co-transported in with a hydrogen ion. And then you can see how the peptidases are working on it inside the cell. So on the outside of the cell, it'll be trypsins and chymotrypsins and so forth. And then it'll be these aminopeptidases and stuff internally as well. I'll continue to break it down. Now, smaller peptides can be carried across through transcytosis. So you have endocytosis, that's what you're seeing right here. So a small peptide will be absorbed through endocytosis, then a vesicle will hold it. Remember, that'll be like a cell membrane. Then it's going to be phospholipid membrane. You're going to have your peptide in it. And then ultimately it's going to be transported across the cell, go through the interstitial fluid, and then be absorbed into your capillaries, traveling down to the liver. Now, babies actually do this more than adults. They'll have a bigger chunk of protein. And this is one of the reasons, again, as I was alluding to earlier, why they're more likely to form food allergies if you present certain foods to the baby before it's really ready to digest it. Carbohydrates are going to be broken down too. So the pancreas is also going to secrete enzymes as well as the brush border cells, things like amylases. And some of this digestion may take place in the cells themselves. So remember, the carbohydrates that we could digest include things like starches and glycogen. We can't break down cellulose. Some animals, thanks to their symbiotic relationship with bacteria, can. They can use cellulases and stuff like that. But here you have glucose being broken down to starches and glycogen into what we call disaccharides. Disaccharides include maltose and sucrose and so forth. Then, thanks to maltases and sucrases and lactases, as you see, the ACE means it's an enzyme. And maltose sugar can be broken down into its basic components, glucose. Or sucrose can be broken down into glucose and fructose, and glucose and galactose. So here's how lactase does that. So if you don't have a lactase enzyme, you can't break down lactose. Where do we find lactose? Obviously in milk. So some people have trouble digesting lactose because they are, and so they're lactose intolerant because they lack this lactase gene. Some people lose it particularly after they become adults. Sucrase obviously breaks down sucrose into glucose and fructose. We call those monosaccharides. So we have a long chain, a complex carbohydrate, which is our starches or glycogens. Glycogen is the carbohydrate you find in muscles and livers and so forth in animals. Starches are obviously kind of components. Then we digest them, we eat them. Amylases break them down to disaccharides, and then we ultimately break them down to monosaccharides. You'll see that a lot of these sugars get absorbed through co-transport. So sodium and glucose or galactose will be transported into the cell and then ultimately transported out into the capillary. So sugars are quickly absorbed into the bloodstream. In fact, the brush border cells will use another nutrient instead of sugars, so that it maximizes putting sugar into your blood. I need to double check on which one that is. But I believe the brush border cells are good at not taking that sugar out. Also, see, glucose is absorbed by indirect active transport, coupled to facilitated diffusion on the GLUT2 transporter. And the fructose uses a different transporter called GLUT5. But that's the basics of how sugars are absorbed. And so I'm going to stop here, and then we'll continue on talking about the digestive system. Talk to you later. Bye.